Hello, hello, hello. I don't know what happened there. That was kind of funny. Welcome, welcome, welcome to the stream. Hello, hello. Hello, The Wolf. Hello, A. Hello, Dark Cube. Hello, More Juice. Welcome to the stream. Hello, Slipta. It's alright. Glad that you've come around to say hi. Today, I am going to be doing the first of my Biology 101 series, which is going to be very, very exciting. But before, as we just let people file in, I'm going to tell a little story. It's been really warm and very beautiful outside these last couple days, and Toxo has convinced me to apply sunscreen, as you should when the sun is out, or just in general, if you are going to go out, you should be applying sunscreen. You actually should be applying more sunscreen than you even believe you should. But I had always kind of avoided this because I don't like the sensation of rubbing creams on my skin. It just feels kind of bizarre to me. But then something unrelated happened in terms of sunscreen appliance, and that was at our graduation dinner, where I get to see the generation of students that I have, taught this course for graduate. During this ceremony, I had been mistaken for an undergraduate, and I thought that that was very nice, considering how old I actually am. And I thought to myself, I don't know when, but eventually one of these days is going to be the last day I am mistaken for an undergraduate student. And I thought that is that's going to be a very sad day. So I came to Toxo, and I very sheepishly sort of admitted that I would like to take on more of her skincare routine because, I would like to push that day when I am unambiguously not an undergraduate student far and far away into the future. So please, to you all, if you want to hold on to your visual youth, put on sunscreen when you go out. That is my task for you. There will come a last day when you get carded too. Yeah, that's already starting to fade in terms of how often I used to get carded. But I'd like to keep that as far ahead into the future as physically possible. People will start to refer to you as sir. The funny thing is that because I actually don't have a doctorate, I am called by my first name in class. So to me, that feels like I am younger. I think carrying the title of doctor is quite heavy. All right, so let's take a look at this poll, and we'll see that of the current poll filler outers, we've got a pretty even split between high school or less or a required class in college, which is very exciting. We've also got a biology professional somewhere in the mix, which is also very cool. Depending on that level of biology professionalism, I am hoping that I can still provide something of interest. So today we are going to be talking about DNA. I figured it would be fitting to start with DNA because DNA is the first step of the central dogma. This is the idea that the flow of information in biology flows from from DNA to mRNA to protein. And so instead of maybe starting from a historical perspective, I thought it would be interesting to start at a theoretical beginning. All right, so let's get to this presentation. So for those of you who don't know, this title, extremely beautiful, shockingly brilliant and cool, I have these letters highlighted because each one of these letters represents one of the nucleobases found in the structure of DNA, which we will get into very shortly. Alrighty, so let's begin. So in 1869, funny number. Oh, I should mention before I get too deep into the presentation, the way that this is going to work is not like a typical university lecture. This is more for engagement of science, so more for your entertainment than it is for test taking. So I don't have everything about DNA here that you would need for a first year course, of course, but it's more like to train you to think critically, as the scientists of the day did when they themselves discovered DNA and the properties by which DNA has, which I think is a very interesting story because there's some pretty elegant biology that we're about to see in this presentation. So we start at the beginning in 1869. Friedrich Meischer. I should have looked up these pronunciations beforehand. Apologies if I mispronounced these. This person accidentally purified DNA, complete accident, right? So what happened was that this cultured scientist wanted to study neutrophils. So I am a neutrophil, so this has a very meaningful factor in my history at the very least. And in the study of neutrophils, this person wanted to separate the nucleus, so wanted to purify the nucleus from these noteworthy, very cool cells. So in order to purify it, he had worked out a protocol where he first added a base and then added an acid, which is actually what we currently do in terms of what real biologists would do to purify DNA. So for any biology professionals in the audience, this is the basis of the protocol for the mini prep, a very common lab technique to purify DNA. However, what he didn't really expect was to get this weird ass gunk. If you've ever purified your own DNA or DNA from strawberries, you'll notice that DNA, when it is purified, one of the steps during the purification will often result in the precipitation of this snot-like material. So this is a weird ass gunk. Now, Friedrich didn't really know what this substance was, but since it came from the nucleus, he called it nuclein. And so at this point in time, there was no general idea of what DNA was, or even how genes or evolution, I guess Darwin, I think, had already finished by this point. So evolution was sort of known. Mendel's work was lost. So the idea of inheritance at this point is still very primitive. So this, just for the time being, was a very cool, but maybe not that noteworthy discovery. Because this substance was new and had no obvious function, people were beginning to think about what this nuclein could do. And so the stage was set much later in the early 1900s by Darwin and Mendel. So Darwin being the founding father of evolutionary theory with his book On Origin of Species, and Gregor Mendel, who did his work in pea plants, showing that there were set rules for certain traits to be passed on from parent to child, doing his work in the pea plant. And so there was this general sense, this was beginning to overturn a lot of what was commonly known or believed in biology, that organisms were static. We now know at this point in time that organisms were not static, that inheritance and evolution were actually a thing. And so the race was on to find what was responsible for communicating that information throughout the body. I remember my first DNA lab, seeing it precipitate made me feel similar to seeing code printed out on paper. That's really interesting. It's kind of meaningful. If you are the type of person to be, what's the word? Not sympathetic, but as someone who sees meaning in things, we'll say, seeing your own DNA can be quite an experience. In 1929, this dude, Phoebus Levine, this guy looks a lot like Richard Dawkins, it's almost uncanny, figured out the chemical composition of DNA. So he found that DNA was made of these segments, not segments, but of these pieces called nucleobases. And so he actually had found that DNA was made of these repeating units, a string of nucleotides. One of them is shown down here. This one is adenine, A for adenine. And put together the fact that a nucleotide is a phosphate group, that's this guy right here with the 4 O's, attached to a deoxyribose sugar, this guy down here, bound to a base, which is this ring structure up here. So this is what is called a nucleotide. And all DNA is 2 strings of these nucleotides wound up around one another. Now, while Phoebus here did successfully understand what DNA was made out of, he got some details of the actual structure of DNA wrong. He believed that there were only 4 nucleotides per DNA, and thus this was too small to be a carrier of genetic information. Welcome, Surprise Pants, to the stream. Phosphate group sounds OP. I agree. There's a lot of negative charges here, which actually does have some relevance for what we're going to talk about when we talk about the structure and chemical properties of DNA. So why is this called a deoxyribose sugar? This is more of a fun fact if you are not majoring in biology, but down here, this is what's called the deoxyribose sugar. In a ribose sugar, there's supposed to be another OH group on this carbon. So when you see mRNA, you'll know that RNA does have that oxygen here, making that a normal ribose sugar. Welcome to Dyslexic Demons. Welcome Clockwork to the stream. Hope you're doing well. We've just started talking about the history behind the study of DNA. I just finished my intro to genetics class, and genetics is so much more complex than I could have imagined. It's true, and we're only going to scratch the surface here. We'll have a whole dedicated lecture to an introduction to genetics in the future. So the thing about DNA is that DNA was purified and studied before people even knew what DNA was. So for a lot of cell biology's early history, proteins reigned supreme, right? At this point in time, at the point of Phoebus, DNA was just some weird goop inside the nucleus of cells, not really obviously useful in any kind of way. But in terms of the early beginnings of molecular biology, proteins have been already proven time and time again to be important thing doers. They do things. Proteins are really important and very, very useful. And at this point, it seemed like the life of a cell revolved almost entirely around the functionality of these proteins. And so many people were quick to write off DNA as just some weird structural element, like it didn't really do anything, but it was maybe there to help proteins do something. And so there was some resistance to adapting a belief that DNA could be the molecule of inheritance because proteins were already proven to be very useful. In 1927, and this is crazy, Nikolai Koltsov predicted the role, function, and mechanism of DNA before any of the other names that you would typically find in a biology textbook did anything. So this dude was a straight up time traveler. Because this insight came before a lot of the upcoming pivotal experiments determining what the function of DNA was. So this is a quote from Wikipedia. In 1927, Koltsov proposed that inherited traits would be inherited via a giant hereditary molecule, which would be made up of two mirror strands that would replicate in a semi-conservative fashion using each strand as a template. And unbeknownst to the rest of the world, this guy got it right even before the experiments would prove him correct. This was a spicy hot take made 20 years before Watson and Crick. So for those of you who don't know, Watson and Crick are commonly given credit for elucidating the structure of DNA. So that just gives you an idea of the fact that this guy was so ahead of his time. Big brain time. Yeah, bro, how? Yeah, I don't know. That's why, like, you know, I'm not a historian of science, so I didn't have the time to look very deeply into just how this guy came up with this idea. But maybe some insight comes from this thing that he had wanted to coin called modus molecula ex molecula, molecules coming from molecules. Maybe he mathed out, or logically mathed out, that this could be the only way that biological information could be passed down, molecules coming from molecules. And like, this dude's gaze in this portrait, dude, he sees the future. I mean, look at this piercing gaze. He knew and was so ahead of his time. Unfortunately, didn't get to live to see him get credit for it, unfortunately. Yeah, exactly. This was before people figured out the double helix. Absolutely insane. Okay, so this was a time when people were searching for the molecule of inheritance. What carries genetic information between your parents and you? And so there are some aspects of DNA that make it a plausible carrier of genetic information. Welcome, Craft. Not very late at all. We just learned about a time traveler who had predicted the structure of and function of DNA 20 years before Watson and Crick did, which is just insane. So we're looking for the molecule of inheritance, and DNA has some things going for it, right? It has a string of 4 repeating units. So I know Morjuice here, I believe I learned, was a software developer. So this 4 repeating units is very reminiscent to bits, zeros and ones that your computer code is made out of, or rather how your computer works at a very basic level. And so the fact that there's this repeating set of information makes it a decent candidate for carrying genetic information. DNA is also at this point known to be massive, right? So there's enough of this particular molecule to code, potentially, for complex information. However, at this point, people didn't really know what it did, but that also makes it good because people didn't know what it did, because maybe what it actually does is have this capacity for carrying information. But at the time, it's hard to unremember that DNA is genetic, because protein has a great candidate, is also a great candidate for conveying genetic information as well. Instead of 4 repeating units, you have 20, sometimes 21 repeating units. So there are 4 bases, A, G, C, and T, but there are 20 to 21 amino acids, depending on what you count as an amino acid. And so that potentially allows you to, I mean, I don't know a lot about how computers are built, but I'm guessing this is a lot more complex, or maybe you would be allowed to do a lot more with computation if you had 20 base units instead of just 4. Proteins were also proven to have a lot of different functions. Proteins are responsible for doing the stuff that your cell needs to do. And they also come in very unique different forms. If I show the chart, I'm not entirely sure what chart you are referring to. And again, protein has proven functions. So something fortuitous happened in an experiment that was, at the time, not thought to be related to genetic material. At least in the conception of this was maybe not pointed at understanding inheritance. So Frederick Griffith was making a pneumonia vaccine in 1928. Oh, the amino acid alchemy looking chart thing. Unfortunately, I don't have that pulled up. We're actually going to do that in the protein lecture, so bear with me. DNA hasn't shown functions yet, not at this time. So 1928, they had no idea what DNA actually did. It was just some snotty thing that was locked in the nucleus. But in 1928, Griffith was making a pneumonia vaccine, which, seems to be unrelated to inheritance. And what was known at the time was Streptococcus pneumoniae had 3 distinct forms. The type 3S Streptococcus was virulent, which means that if you were to inject this into a mouse, it would die. It causes disease. And type 2R was not disease-causing due to a lack of a protective membrane. So the type 2R, the type 3S has this kind of membrane wall that surrounds it, that shields it from immune systems, from the immune system from clearing it out. So there are 2, for our story here, 2 relevant types of Streptococcus. And what he observed was that rough bacteria, this type 2R, can transform into the smooth bacteria. So smooth because it's encapsulated in this smooth shell. I used the word shell in my Plague Inc. short and gave Plague Inc. shit for it, but, bear with me. So one type of bacteria can somehow transform into the other type of bacteria. And this was interesting. The question is how? How would you attempt to figure out the mechanism? Does anybody have any guesses? Try not to, if you're a biology student and you already know the answer, try not to spoil it for everybody else. But you see that there are 2 distinct types of bacteria, and one bacteria can transform into another. What's a hypothesis that you might have about the relationship between these 2 types of bacteria? Now, it doesn't have to be correct, but I highly encourage you to try, because even if you try and you say something in chat and it ends up being wrong, if you force yourself to come up with an answer, that's a really great workout for your brain, because what will benefit for you in your future education, if you're still in school, is practicing coming up with new ideas. So I'll give you a little bit of time to think about how one bacteria might transform into another bacteria. And it can be as abstract as you want, right? You don't have to have a concrete answer backed up by science. You can think a little abstractly if you don't have all the pieces. Vegacraft, they are the same, same DNA, but different gene expression depending on environment. Yeah, that is one possible hypothesis that could have been true. So maybe there's an outside signal. And actually that's very relevant for when I talk about bacteria. This idea of quorum sensing is the fact that bacteria can actually sense molecules in their environment that are messengers from other bacteria to allow them to adapt to their current condition. The smooth strain bacteria are virulent, which means they are capable of causing a harmful infection. Yes, so to clear that up, because they have an additional membrane, the immune system can't effectively kill them, which means they can go off and kill whatever it is that they're infecting. All you gotta do is take off your rough exterior by crying at sad movies. That is true. That is true. You know, we always ask how do we get rid of bacteria, but we never ask how the bacteria is. They have something that can change. I'll take that answer, eh? They have the capacity to change. And so if we're putting down a hypothesis, if we're trying to figure out what causes a bacteria to change, we might suggest that there's something in the rough, so that the smooth bacteria makes that can convert the rough bacteria to smooth. That's one potential hypothesis. LPS layer, I believe that might be correct, although I don't know the particular details for this bacteria. Do they have a way to compare the nuclein goop between the two strains? That's a great idea. At that time, I do not believe that sequencing was a thing, so there was no way to actually see whether or not their nuclein was different. The bacteria got in a relationship, and that's what caused it to change. You know, that's actually the closest thing that we'll get so far. But Izzy, you are right on the money. So one way that this could happen is maybe the smooth bacteria are giving the rough bacteria something that causes the rough bacteria to become smooth. Right? So how would you go about testing that? If your model was that there's something that S makes that converts docile to S, how would you go about testing that? Horizontal gene transfer, that's a little bit of a spoiler. I won't explain what that is. But yeah, you are correct. But not for the purposes of this experiment. So if one bacteria makes something that converts the other bacteria, how might you test that? Well, one way that Griffith came up with was to kill the S bacteria with heat and give their guts to the R bacteria and see if the R bacteria become S and kill mice, right? So bacteria make a whole bunch of stuff. Sometimes they spit that stuff out into their environment, but sometimes they keep that stuff inside. And so if you want to test whether or not one bacteria makes something that can convert another bacteria, pop that bacteria open and then donate the guts. Isolate the samples and see if transformation happens only if it's one strain present. That's actually a really good idea, and I believe that they have shown that you need the presence of both bacteria in order for R to become S. Damn, that's kind of hardcore. Yeah, and we'll see in a little bit why I think this was fortuitous in a way, where this is kind of a hardcore approach, because I don't know at the time whether or not they had the French press technology. For those of you who don't know, you can use a French press, although it's a fancy lab French press, to crush bacteria without burning them, because heat killing them can destroy all sorts of different things. But I thought this was actually kind of nutty, because what if this thing that caused R to become S was destroyed by heat? Then you would not have captured the thing that turns one bacteria to another. So I think, it's kind of fortunate that this ended up working, but I could foresee a timeline where this was not the case, right? Molecular coffee, exactly. So this is what the Griffith experiment is, and it is the first major experiment to show that there is a molecule that can pass on genetic information. So in one set of experiments, Griffith injected these mice with the rough strain, the non-virulent strain, and you would expect that the mouse lives. He then injected these mice with the smooth strain. And so the smooth strain is the virulent strain. And if you inject the mouse with the oh god, I can't speak. If you inject the mice with the virulent strain, the mouse dies. This makes sense. That's what I set up this experiment with. Nothing too surprising here. if you kill the smooth bacteria, so this is the heat-killed smooth strain, and you inject it into the mouse lives. That's not necessarily obvious why that would be, but it certainly makes sense, right? If you kill the bacteria are not going to be actively bacteria, and so the mouse should live. So, chat, what do you think happened when Griffith added the heat-killed smooth strain with the rough strain? What happened? This fourth panel that I'm gating. You add the heat-killed bacteria to the rough strain. Does the mouse live or does the mouse die? What is your prediction? Yep, dead bacteria not being bacteria makes sense. I know. That's what I love about biology, is that a lot of things can be broken down to logic as simple as that. Mouse died. Exactly. Great job. So if you add this heat-killed smooth strain to the rough strain, something happens. The rough strain alone is not enough to kill the mouse. The heat-killed smooth strain is not enough to kill the mouse. You combine the two, and all of a sudden the mouse does die. Do they somehow combine and kill it? Exactly. So I love that agnostic approach, right? If you don't know the answer ahead of time, this is what makes this so cool, is that there's something happening between the heat-killed bacteria that normally causes disease and the normal non-virulent strain. So very cool. So this Griffith calls the transforming principle. There's something in this heat-killed smooth strain that transforms the rough strain and allows it to kill the mice. Very cool, very cool. Smooth bacteria gave something to the rough one. Exactly, and we want to know what that is. There's something that the smooth strain can make that can transform the rough strain into the smooth strain, so this is called the transforming principle. The thing is that because Griffith used heat, it's actually an incredible stroke of luck that the DNA that the transforming principle was not destroyed in this process, right? Because Griffith actually heated the bacteria up to 60 degrees Celsius to kill them. And a lot of proteins will actually be destroyed at 60 Celsius. But DNA is actually very, 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 very stable. You can leave DNA, purified DNA, out at room temperature for a very long time and it won't degrade, which is unusual for a biological molecule. Now, of course, the melting temperature of DNA, the temperature by which these two strands will separate and become non-functional, depends a lot on the length of the DNA piece, the salt level, and the nucleotide composition. But generally, DNA will melt between the temperatures of 50 degrees Celsius to 100 degrees Celsius. So there are pieces of DNA that you can boil and it still won't denature or fall apart. And so I wanted to talk a little bit about what makes DNA so stable. So this is a 101 kind of lecture. And while this isn't really a chemistry channel, I think it's kind of cool to see DNA at this level of detail to kind of understand a little bit of it as to why it is so strong. So let's take a look here. These are the nucleobases, A, G, T, and C. Adenine here has this chemical structure. We have the phosphate backbone. So if you don't know what you're looking at, this winding backbone, this red bit, and this blue bit that winds down the entire length of the DNA, that's this part, the phosphate ribose sugar. So this is this. The bases, these ring structures, that's these two. So in this particular example, we can say that this T represents this yellow bit, this A represents this green bit, this C represents this purple bit, and this G represents this blue bit. And so part of why DNA is so stable, or how DNA can become so stable, is these hydrogen bonds. So it's okay if you haven't taken chemistry yet, but if you just observe these lines are indicative of hydrogen bonds forming. And you'll notice that when G and C bind together, they form 3 hydrogen bonds. So the more GC pairs you have in DNA, the more hydrogen bonds you have, the tighter these strands of DNA actually stick to one another, which is very interesting. Is longer DNA more stable than short DNA? Exactly. It is more stable than shorter DNA. There's just more stuff that you have to tear apart. I hope my DNA wouldn't melt at room temperature, otherwise I'd be screwed. Yeah. Pretty cool that we adapted life in that regard. All right. Aren't hydrogen bonds weak? That's a great question, A. The thing is that there are a ton of hydrogen bonds between strands of DNA, right? And there are 3 of them per GC pair. So the longer the length you have, the more of those hydrogen bonds you have. That's not to say that hydrogen bonds are the weakest. They're certainly not the weakest chemical bond, but they're not the strongest either. But good enough to keep DNA intact. But that's not the only reason why DNA is so stable. You actually have this other force called van der Waals forces that are in between these bases. So not only do you have these hydrogen bonds that bind this side of the ladder to this side of the ladder, these bases, the T and the C here in this cartoon, these have a very, very weak force that keeps them together as well. This kind of pseudo-gravity, I'll say, although physicists and chemists might get mad at me for calling it pseudo-gravity. But yeah, I don't want to get too deep into how van der Waals forces actually work, but just know that because these 2 molecules are close to one another, they exert a little bit of this van der Waals force. And the thing about van der Waals forces is that they are additive. So the more van der Waals interactions you have, the stronger they get. So there are these tiny, tiny, tiny forces in between the rungs of the ladder. But across a long stretch of DNA, those forces actually really help keep this molecule stabilized, which is very interesting. Van der Waals is the weakest, exactly. Very good, A. Van der Waals is the weakest. One of the things that really broke my brain in undergrad was realizing adenine was basically ATP. That's true. So for those biologists out there, or for biology students out there, this A, it has 1 phosphate, but actually if you add 2 more phosphates on here, that's actually ATP. That's the cellular fuel. And indeed, you actually burn ATP to stick this adenine inside the DNA ladder when you are making DNA itself, which is very cool. So you have the hydrogen bonds that are in between the bases. You have the van der Waals, which interacts along the rungs. You also have something called the hydrophobic effect. So there's water surrounding the DNA, and there's a force by which it is not very efficient chemically, energetically, to be solvated in this water. So I'm not going to get too deep into this. We'll talk a little bit more about this in the protein lecture, but basically, water doesn't want to be in between these rungs, which is why the water prefers to stay on the outside. You'll see, and Surprise Pants kind of alluded to this, one of the many properties of DNA is that the backbone is charged, and that's because this phosphate group with all these oxygens and all these electrons loves to bind water. But these guys in the middle, not so much. So the water would prefer to stay on the outside, which helps keep the DNA molecule together. So Griffith got kind of lucky because Griffith killed his bacteria at 60 degrees Celsius, but actually DNA is so stable that it would not have melted. If you imagine an alternate universe with different rules, maybe he would have destroyed this accidentally, and then molecular biology would have been set back 20 years. So we're really setting the stage for this showdown. Is the transforming principle protein or DNA? We still don't know yet, by the way. We just know that when you add the heat-killed smooth strain and the rough strain together, that the mouse dies. We still don't know what the transforming principle actually is. So this experiment, Oswald Avery, Colin oh, that's not 1994. That's way too late. I think that's 1954. That should be. That's a typo. Avery, MacLeod, and McCarty showed that the transforming principle was likely DNA. And this experiment was heavily based on the Griffith experiment. So it's actually almost the same thing, except they were a little more clever about how they set this experiment. Oh, sorry. How they set this experiment up. Okay. So I just kind of recreated this particular setup. So we have the smooth virulent strain. You pop it open, you add it to the rough strain, and you get a dead mouse, right? That's what the Griffith experiment had shown. So what Avery, MacLeod, and McCarty wanted to do was narrow down the possibilities. So they wanted to ask the question, is the transforming principle DNA or protein? So how would you take this basic experimental design and craft it in a way where you can distinguish whether or not the transforming principle is DNA or protein? And it's okay if you don't have a biology education. If you are a biology student and you know the answer, please keep it to yourself, at least for a little bit, as we allow the non-experts to try their hand at thinking like a scientist. How would you distinguish whether or not it is DNA or protein that conveys genetic information. How would you do it? If you were given this experiment and you were asked to refine it, how might you determine whether or not it's DNA or protein? Gonna go ahead and hydrate. You should too. All right. Looks like A has an answer here. I suspect that you have taken classes because I think A, you've been on top of some of this for a while. So A says destroy only protein or destroy only DNA. And that is exactly what they did. So if you add enzymes that destroy protein, good job, Dark Cube. You also achieved the correct answer. Maybe I should wait a bit longer for my chat to come up with answers. It's a little hard to tell because I don't know how much lag is after when I speak. But yeah, so good job. That's a great guess. That's why I love doing these lectures so much, because people think science is hard, and it is. Because science is hard because you need to know a lot. But actually, the logical aspect of science is very teachable. You don't have to be like a super genius to figure some of this stuff out. And in fact, my personal taste and you know, my V theorem partner Sidere has a different taste in science than I do. My taste is science that makes you think, damn, I wish I thought of that. Her taste in science is more revolutionary, I'll say. Like, what appeals to her is, and I could be wrong, but this is what I'm getting, is that what appeals to her is when things are overturned, when new things are learned that overturns what old things were, which I think is very cool. And it's fun that we have like different tastes in science, because my taste is very much like a beautiful experiment like this, something that is so simple that anybody could have thought of it, but they didn't. That, I think, is the coolest science. Damn, I wish it was me. Exactly. Like, I wish I was smart enough to have, or I wish I had been in a place and time and had the insight to come up with this. Because the reason why it's brilliant is because anybody can understand. Anybody can actually do it mentally themselves. They can figure it out, but they didn't. And that's where the brilliance lies. I abstained because I knew. Yeah, that's fair. Size of Science Rebel. She is. Just got here. What did I miss? Welcome, Thomas. We're talking about the transforming principle. So we are, together with chat, figuring out how Avery, MacLeod, and McCarty figured out that the carrier of genetic information was actually DNA. So, destroy protein, there's a dead mouse. And if you destroy the DNA, the mouse is alive. So it seemed like the thing that is turning the rough, non-virulent strain into the deadly, virulent strain is DNA, because when you destroy the DNA, the mouse is alive, right? This is pretty amazing, right? This nuclein, this snot that we found in the nucleus that we didn't know what it did this entire time, that turned out to be one of the most important molecules to ever exist, right? Just insane. Does bacteria usually take random DNA in their environment and create stuff from it? Bacteria can uptake DNA. It doesn't always result in making stuff with it, but bacteria like to pick up sequences from their environment. That is definitely true. How did they selectively destroy protein or DNA? That's a great question. So at this point in time, biology and chemistry was advanced enough to purify these enzymes called proteases. So I believe the protease they used was both trypsin and chymotrypsin. These are very common enzymes. I believe they're actually in your gut as well. These proteins just chew up, these enzymes just chew up protein. So that's how you destroy protein. Now, you're kind of getting ahead of me, more juice. At the time, they didn't really have a great way to destroy DNA. They haven't really isolated an enzyme that just does that, but they got what was called biologically active preparations of samples. So they found stuff that they can get out of cells that would destroy DNA. They didn't know what that stuff was. They didn't purify it, but they had stuff that could destroy DNA. And so that's what they used. And the thing is that your instinct to wonder how it is that they did that is exactly why there was pushback on the Avery, MacLeod, and McCarty experiment, as you'll see in this slide. So the AMM experiment wasn't actually sufficient to quell the haters, right? Some believe that the structure of DNA was not complex enough. As I mentioned earlier, Phoebus believed that the size of DNA was just 4 nucleobases per molecule, so there's no way. You can't write a code for life and have it only be 4 lines of code, right? That's just not feasible. Phoebus also thought that the DNA was just a structural component of the chromosomes and that protein was a transforming principle. The methods were somewhat crude, as more juice you had prompted me to talk about, and they could not completely rule out protein contamination. So that was the issue that these haters had. That because they used this extract, this unclear, messy, not pure extract that destroyed DNA, but they had no idea how, that prompted there to be skepticism. And it's easy to laugh at them now because we have hindsight, but at the time, protein was the big thing. And so this other thing, it was just so hard to believe that DNA could actually do anything. Dab on them, exactly. There's going to be an experiment that dabs on the haters in just a little bit. And that dabbing on the haters came in the form of the Hershey and Chase experiment. And so Hershey and Chase deliver a big L to the haters, and I think most people can agree that after Hershey and Chase, there was no argument. DNA is the transforming principle. So we are once again going to inhabit the space of these scientists. I'm going to give you what Hershey and Chase knew, as they were both scientists that worked with phage, as you can see on this slide. I'm going to give you the information that they were aware of. I'm going to have chat come up with the experiment to help them determine what it is that determined the code for life. Ready? So they used bacteriophages and bacteria to show that DNA is the genetic material. And they took advantage of the fact that proteins and DNA are comprised of distinct elements. Proteins, if you just break down proteins, elementally what they are a mixture of carbon, hydrogen, nitrogen, oxygen, and a little bit of sulfur. DNA are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, so bacteriophages are viruses that only infect bacteria. And so chat, given what you know about how viruses work, this is enough information to intuit how Hershey and Chase figured out that it was in fact DNA and not protein. And again, if you're a biology student and you know the answer, please give us a little bit of time while I vamp while you think. This virus only infects bacteria. Protein and DNA are made of chemically distinct compositions. And so I want you to come up with a potential experiment to figure out how you would distinguish which of these is the carrier for genetic information. And again, just take a stab at it. You don't have to worry if the technology is there or not, if it's possible to do or not. Just abstractly come up with something, and we'll see what you can find out. Check for sulfur or phosphorus in the virus. Megacraft, that's very, very close. It's very, very close. You are eyeing the correct answer. Phosphorus, yes, we're picking up that there are differences between these two. So we definitely want to look at phosphorus or sulfur, phosphorus and sulfur. So I want to let this stew a little bit, because you've identified, Chet, you've identified what we're looking at, what our attention should be on. But can I push you to think a little more carefully about how you would check for this, or what you would want to see? So check for sulfur or phosphorus in the virus is almost correct. It's not quite correct though. Do we want to check for sulfur or phosphorus in the virus? Come on, Chet, I believe in you. Megacraft's answer is one word away from being completely correct. One word away. Where do we want to check for sulfur or phosphorus? It's not in the virus. Viruses, they infect bacteria, right? And in order to infect that bacteria, exactly, yeah, Megacraft, you got it. So not in the virus, but you want to check the bacteria. Very good, very good. And so how might that allow you to determine whether or not the transforming principle is in DNA or protein. Let's take a look. So this is the relevant experiment here. You can radio label sulfur and you can radio label phosphorus. So you know, like, there are isotopes, and isotopes of elements have certain properties like radioactive decay. And so you can radio label sulfur and you can radio label phosphorus. So you can track the location of sulfur and phosphorus containing molecules. And so what the Hershey and Chase experiment did was that they labeled two different types of bacteriophages. One of them they labeled with sulfur, and the other they labeled with phosphorus. So in red, you see that the protein portion of the bacteriophage has been labeled. And on the right, we see that the phosphorus portion of the virus has been labeled. And so during bacteriophage infection, the phage will stick onto the virus, inject DNA, and then the capsid will fall off, because what the bacteriophage cares about is getting the genetic material inside the bacteria, and then that genetic material does all the work for this particular bacteriophage. And so what they had found was, after infection by either one of these, when you take a look at the infected cell, do you see red or do you see green? Well, it turns out that you actually see green. You don't see that red. So what Hershey and Chase had found was that the protein portion just temporarily sticks on that cell. It doesn't enter the cell at all. But the DNA, the part that we labeled because it contained phosphorus, that sticks around in the cell. So it could not have been the protein portion. It had to have been the DNA, because the protein portion doesn't enter the cell at all. Labeling is like making it detectable. Exactly. So we can detect the presence of the DNA molecule in the bacteria, and the fact that DNA is the thing that is inserted in combination with the Griffith and McAvery experiments, all of those combined provide this definitive proof that DNA is the carrier of genetic information. Very, very cool. Logical. Exactly. That's where I find classical molecular biology to be so beautiful, because you don't have to learn about like a billion different techniques. This is logical, it's reasonable, it's easy to explain, and again, it gives you the feeling of, damn, I wish I thought of that. So bacteriophages are virus. They make bacteria make viruses. This requires instructions to make viruses. Since protein is not injected into the bacteria, it can't be the protein that contains the instructions. Combined with the Avery-MacLeod-McCarty experiment, this conclusively proves that DNA is the transforming principle. Genes are made of DNA. Awesome. Once the structure, or once the function of DNA was determined, the world's attention turned into the structure of DNA, because structure informs function, form follows function, or function follows form. And so learning what DNA actually looks like is a big part of this history. But I'm actually not going to go too deep into this, because I'm not a structural biologist. And so I'm not qualified to talk about how this was put together. And there are plenty of other sources that recount the Watson-Crick-Franklin drama a lot better than I can. But let's just say all three of these scientists worked very hard to come up with our understanding of how DNA actually looks. Okay. So, don't some bacteriophages also inject proteins? That's a good question. I'm afraid I don't have an answer for that, why they didn't see any protein in their cells. Maybe it's a unique quirk of the particular bacteriophage they picked. I'm not going to go too deep into Watson and Crick's contribution, but just know that they are commonly given credit for ultimately putting the entire picture together. But I want to take a look at the Rosalind Franklin contribution, because to me that is the more, I guess you could call it, spiritual element of all of this. This is an exhibit called, I believe it's called Photo 51 or Experiment 51 or something. So Rosalind Franklin was a very talented X-ray crystallographer. So this is a scientist whose job it is to create crystals of molecules to identify what they look like at a molecular level. And so I don't know how to go from this blur to the structure of DNA here. I was taught how this happened in grad school, but to be honest, I failed that chapter. I didn't need to do that very well to pass my grad school courses, so I never actually figured out how to read a picture like this, but just know that through some very clever and potentially difficult structural biology, you can go from this grainy photo into figuring out exactly what DNA looks like, which ends up being like this. So you can get very precise detail with X-ray crystallography, including the number of turns or the space, the length of the DNA ladder and how often it turns. I wouldn't be able to tell you how to go from this to this. Maybe you go to grad school for that. How could you find something from this image? Dude, structural biologists are wizards, man. I don't know how they do what they do. I could never figure this out. But they can, and it's crazy. This picture's from Wikipedia, by the way, so I can't take credit from this. If I drew this, then I would know how it works, but I didn't, so. So what do we know about DNA now? So I wanted to tell that story because it's a couple of experiments that are very easily intuitive. Not easy, but they're straightforward and intuitive. I wouldn't say they're easy, but anybody, when given the right information, could potentially figure it out, right? Or be led to figure it out themselves. It doesn't have to be, you don't have to coach them too much to figure that out. Is it like looking at DNA from top down or something? I just want to correct that real quick because it's not quite right. It's looking at scattering through a crystal. And again, I don't know exactly how it works. Go to grad school and you'll find out. So what do we know about DNA now? I'm transitioning and skipping a whole lot just because I don't find the experiments that lead up to the current day to be as compelling as those pivotal experiments. Not to say that they're important or interesting, but this is a 101 introduction to the concept of DNA. So let me go through just some basic stuff that you would learn in a 101 type lecture so that we can end the presentation on something from the modern day, which is ultimately my goal. So we know at the very basic level that DNA, the bases are comprised of 4 different types of bases. There are the A's, there are the T's, there are the C's, and there are the G's. And as you saw earlier from my structural biology bit, not the X-ray crystallography, but before that, that there are a different amount of hydrogen bonds between C and G, T and A. These nucleobases form the rungs of the DNA ladder, which form this shape that we have elucidated from Rosalind Franklin's X-ray crystallography. Inside the cells of eukaryotes, these pieces of DNA are wrapped around proteins called histones. So these histones are a key structural regulatory component that form these nucleosomes. So a unit of histone and DNA is called a nucleosome. These nucleosomes are then themselves wrapped up into a thread, and that eventually is wrapped into a chromosome. So it's this coiling upon coiling upon coiling upon coiling, which is why you can fit so much DNA in a single chromosome. These chromosomes, of course, live in the nucleus, which then, of course, lives in the cell, right? Why does RNA have uracil instead of thymine? Why is always a tricky answer to answer in biology. I couldn't explain why. That's not something that I'm equipped to answer at this very moment. It's a good question, though. Interesting one. So that's what we know about where DNA lives and how DNA is structured inside a eukaryotic cell. What we know now, after the many experiments, that DNA is the structural component of genes. So within this chromosome, within a string of this code, lies a gene which can range from anywhere between 1,000 base pairs and very short sequences to like 1 million. Yeah, 1 million, right? That's 10 to the 6. Imagine having to unwrap that. It's true. It would be a nightmare, but your body does it all the time, which is very interesting. So each gene, a gene basically refers to a portion on the chromosome that codes for a certain kind of function. It's actually wrong to say that a gene makes a protein, because not all genes make proteins, believe it or not. Sometimes genes make functional RNAs, which is why there's a common misconception that because most of your DNA does not code for protein, that most of it is junk. That's simply not true. A lot of DNA's importance is structural and regulatory, and you don't always make proteins that do stuff. RNAs do stuff as well. So just wanted to clear up that misconception here. So there's no conversation about DNA without discussing the central dogma. Indeed, I did start the presentation with the central dogma, but the information flow within biology, for the most part, is DNA to mRNA to protein. And so this up here is a pretty complicated graph, but I'm going to explain it just so that if you were ever curious about how DNA works, you'll have a bit of an understanding, hopefully after this lecture, right? So within the DNA, this is like an example of one gene, right? So we're going from one gene, and this is kind of the building blocks of one gene in a eukaryote. Bacteria work a little differently. This is for a eukaryote. So there are three principal parts of genes. There is the open reading frame. This is what people classically think of as a gene, even though it's not, technically all the way there. This is the part that codes for protein. So there's a stop sequence, a start sequence, and a stop sequence, and this part in red is the actual code that gets read and is made into protein. But a gene is so much more than that, right? There are regulatory sequences that can be upstream, downstream, both of that open reading frame. And if we take a look at the upstream component, this sequence of DNA that is before the open reading frame, you'll see that it has a couple of different parts. So the part that most people are going to be familiar with is the promoter. So the promoter is a sequence of genes that, a sequence of bases that the enzyme RNA polymerase really likes to bind to. And so this portion up here is a structural regulatory element that recruits the enzyme to put it in the right place to start making mRNA and then to start making protein. If I had to come up with a programming, I don't know, I'm not good enough at programming to come up with a programming equivalent. I don't know if Morjuice is still in chat, but I think even though I dunked on Tech Bros in my previous stream, thinking about DNA as being read as code is not necessarily the worst analogy to draw. I can't think of an analogy right now, but this is regulatory. It helps to determine whether or not you should be making a certain protein. Upstream of that is a little bit more of an esoteric biology fact, is that not only are there promoters, but there's also enhancers / silencers that are like way upstream, like very far away from the gene. So not only do you have like, hey, RNA polymerase, sit here and start making protein, please. You also have these sequences that are really far away in terms of base pairs. And so those sequences can act kind of funny in the sense where you see this dotted line here. Sometimes the way that enhancers / silencers can work is that in the structure of the chromosome, you actually have folding. So even though this is dotted here, you can actually have this whole dotted part fold and bring the enhancer silencer a lot closer to the actual gene. So that's why there's some regulatory sequences that are really far away from the gene, because you can fold the chromosome in between, or you can fold the DNA in between, which is why it's that far up. If you're not paying attention, that diagram could be mistaken for a data format protocol in CS. I don't really know what it is you're referring to, but I wouldn't be surprised if, geneticists and programmers have a lot of overlap, obviously. Metadata of the file, yeah, that's not a bad analogy to draw. It helps you read or understand when it is appropriate to make a certain protein. All right. Up here is the 5' UTR, so this is the untranslated region. So this is just a chunk of the DNA that isn't made into protein. And then the 3' UTR, same thing, and a sequence called a terminator. This terminator, it is not the Arnold Schwarzenegger equivalent, but it is a sequence of DNA that causes the RNA polymerase to pop off and stop making RNA. You don't actually run DNA unless it's functional. Yeah. Trying to twist myself into, I don't know, nucleosome here. Trying to come up with an analogy was maybe a mistake. All right. So when that RNA polymerase is recruited by the promoter to make RNA from this open reading frame, you generate a molecule of mRNA. What does the UTR do? It just provides a bit of space in between. It's just like there for structural purposes. You don't always want your promoter to be right up at the open reading frame. Otherwise, you might not get RNA polymerase to stick in the right location. It's a little bit of a buffer. A little bit of a buffer. Yeah, Wolf, we're kind of talking at the high school plus level here. So this is some pretty advanced biology. So when that RNA polymerase binds to that promoter, it'll make that RNA. And so as I said earlier, the terminator, that's where that sequence gives RNA polymerase the time and the structure to pop off and stop. So while the gene is this long, the mRNA that is being made is only this long. Okay? And so this is actually going to be a teaser for my prion video because we're going to be talking about introns and exons, but mRNA, when it is first made, is not actually the final product that is made into protein. Eukaryotic RNA has to undergo post-transcriptional modification. So this is what is called sort of an immature transcript because there's still work to be done to make this into an actual protein. So after modification, you cut out these introns. So there are introns in the open reading frame. These are sections that used to be thought of as junk because they didn't get made into protein, but the introns actually do have interesting properties by themselves, which I will get into in the prion video. Basically, you can kind of think of this as programming efficiency or memory. I'm trying again. I'll never stop trying to shoot out of my depth. But if you think about this, this is the immature RNA. And sometimes not all the introns are cut out. So this is one possible mature mRNA that can be made into a protein. If you don't cut this intron out, then you've just generated a new protein with a new intronic sequence in it. This is called splice variants. So not a lot of people know or remember that mRNA has to be modified before it is made into protein. So, so this is the mature mRNA. This is what will actually be read by the ribosome to make protein. What are introns? Introns are sequences within a eukaryotic genome that are cut out at the mRNA phase. So you'll see here in the DNA that these introns are here. These introns are at first made into mRNA, but they eventually get cut out of the final mRNA before it is made into protein. And it's confusing. It's definitely not intuitive. You would think that across evolutionary time, we would evolve a level of efficiency where we wouldn't have to do that, right? But in actuality, this is evolutionarily adaptation. This is allowing you to make many proteins out of the same sequence. So imagine this. This red mRNA can code for a protein. But if you don't cut this out and we insert that here, you've just made a new protein. In fact, if you don't cut that out and you don't cut any of the introns out, that's another protein. If you cut this intron out and leave this intron, that's yet another protein. You can cram a lot of density in the DNA code by selectively splicing the mRNA, which is pretty interesting. The non-coding sequences also play a part in regulation in genes too. Yeah, exactly right, more juice. And additionally, there are non-coding regions of the genome that act as mutation buffers. So you can imagine that if every base of your chromosome was functionally useful, that if you got a mutation in any location, that could potentially be bad. But there are locations in the DNA where you could get a mutation and it won't be a big deal. Okay. So mRNA is processed into a mature mRNA. And then the ribosome, we love the ribosome then takes that mRNA and makes it into a protein. How does it know where to splice? So there are regulatory sequences in these introns called splice acceptor sites. So these are specific sequences of DNA that can recruit the splice machinery. or to help recruit the splice machinery. And I'm going to talk about it a lot more in the prion video, because prions and splicing, there's a very, very interesting story that I don't want to spoil that I'm currently working on for all of you. Trying to have the prion video by the end of June, but we'll see. So that's basically the 101 of how DNA works, right? But from here, there are so many different additional things to cover, like genetics. Like I haven't even done genetics. That'll probably be a 101 lecture at some point. Genomics, which people like MolBio might be more interested in, using computers to study large swaths of data. DNA damage, repair, protection, etc. Evolution, which every biologist has a working understanding of evolution, but evolution itself is actually quite complex. Genetic engineering, which I covered a little bit in my last stream, and structural biology. And as a shout out to structural biology, I just want to take a bit of time to appreciate how beautiful alternate DNA structural forms are. So we're mostly familiar with this form of DNA, right? This is the classic form of DNA that we see. And if we look from the top, they all have these incredibly interesting, almost like a kaleidoscope situation. I don't know if these other forms of DNA are functional, but the fact that DNA has a lot of conformational flexibility makes it so that you can do some really neat things like origami. You can look up DNA origami. Very cool. DNA has all sorts of interesting folding properties that maybe we'll get into in a future journal club. Life is, in fact, crazy. That's why I love studying it so much. Okay, so this is the part where if you are a in training and you want to push yourself a little bit, if you're a biology professional, this is probably boring to you. But if you are a biologist in training, we're going to talk a little bit about a current paper about the tardigrade's DSUP protein. So the tardigrade is an extremotolerant little bugger that can survive cosmic radiation and come out fine, and actually come out fine without massive damage to their DNA. So part of DNA studies is understanding how DNA can be repaired and how DNA can be protected. I'm actually curious about what the DNA machinery uses for error correction, if it is similar to how CS does it. I will do a DNA repair stream at some point, because it's not something that gets talked about in your 101 classes, but it is very interesting the way that happens. And I'm also not prepared to talk about it because there are a lot of moving parts right now. Gattaca watch along stream when? I think I might. I mean, there's so much content to make. You know, I had this hubris when I started this channel that like, oh, I don't want to do certain ideas as journal clubs because I want to save them from long-term videos. But the more I have this channel, the more I realize that I will die before I finish covering everything in biology. Yeah, so tardigrades kind of shrug off cosmic radiation like it's no big deal. And so you could imagine that if you are a scientist, that you would be quite interested in how the tardigrade protects itself from DNA damage, because protection from DNA damage is how we get closer to a future where cancer is not as big of a deal, right? So DSUP is a protein that was isolated from tardigrades that was shown to be protective. But up until 2019, it wasn't entirely sure. We didn't really know how that happened, really. We just knew that the DSUP protein protected DNA, didn't know how they did it. And so I want to take a look at this wild structure. So this is a predicted alpha fold structure. So they can repair their DNA or not. Yeah, tardigrades can repair their DNA, but DSUP is the protein that prevents DNA damage to begin with. So this structure is freaking crazy. There's a lot of flexibility in this structure. And AlphaFold is actually Google's DeepMind database, where they use AI to predict how proteins fold. Of course, this is not reality, right? We have to make that clear. DeepMind, AlphaFold is not just because it has a structure in AlphaFold doesn't mean that that's how that looks like in real life. We have to be sure that we acknowledge that. But if we take a look at this protein, we'll notice that it's like wobbly spaghetti. These helices here, these are called alpha helices. This is a level of secondary structure that the protein has. So this is kind of like the core that doesn't really move all that much. But the rest of this spaghetti string, that's all looking very, very flexible, which is no wonder why they had to use DeepMind technology to fold it properly, because this looks like it would be a nightmare to actually crystallize. So I'm not going to do a full journal club on this paper because this is a 101 lecture. That's not what this is about. But let's put ourselves one more time in the position of the scientist. You have this protein, this D-sub protein. You know that the tardigrade needs it to protect itself from DNA damage. So just guess how, because we can't do experiments unless we have a hypothesis, unless we have a reasonable guess to test, because that's how science works. So how do you think that the D-sub protein could protect the tardigrade from DNA damage? Take some time, chat. I'm gonna hydrate, but I want to challenge you to come up with just a guess. It doesn't have to be right? It never has to be right on this channel. But if you just had to guess this spaghetti string flexible protein, what's one potential way you could see that a protein like this could protect DNA? Nadia, welcome to the stream. It acts like a shield? That's definitely a possibility. Does anybody else have another hypothesis? It acts like a shield. D-sub wraps around the DNA or the nucleus? Yeah, definitely possible. Compact the spaghetti. Yeah. Protects it from mutations? Yep, that would be the downstream. I'm looking for something a little more physical because I like thinking about molecules, which is why I picked this paper. I'm going to say, chat, that you are thinking along the same lines as the scientists I'm about to talk about. So, acting like a shield, protecting it from radiation. Does it aggregate around DNA to protect it like a shell? So you're all assuming that the answer is already known, but I'm time traveling back to the faraway year of 2019 in the shoes of these scientists. So we'll start by saying that that is your hypothesis. That aggregation around the DNA is a hypothesis that we can test. Importantly, it's a testable hypothesis. So that's what these scientists in 2019 thought to test. They thought, hey, maybe D-sub does something like bind to the DNA to protect it from radiation. They don't know that that's an answer, or that that's the answer, but that is something that they could try to prove, right? And so the way that they did this was through something called a gel mobility shift assay. And what this does is it measures DNA binding complexes. So any biology student out there can tell you what this is. This is a cartoon of what's called a gel. And so DNA, as Surprise Pants has brought up way in the back of the stream, that phosphate group looks OP. That phosphate group has so many electrons on it that it makes DNA a very negatively charged molecule. So if you load DNA into a gel and you shoot electric current through it, and you have one pole be the negative end and one pole be the positive end, because DNA is so highly negatively charged, it'll be shoved through the gel. And the thing about DNA is that if you can imagine the gel as sort of like a matrix. So if you are traveling through a gel, the bigger you are, the slower you travel through that gel. So if you are a tiny DNA fragment, you're going to travel very quickly, and you're going to end up at the bottom of this gel. Electrophoresis, exactly correct. More juice. It's hard to word while thinking so quickly. Yeah, that's true. Part of the challenge, I guess, is that I'm still making content. I'm not actually in lectures, so there are chatters a bit faster than you to the punch, but that's okay. I acknowledge that you also had the same idea. So here's the next question that I have for you here. If you have a DNA fragment, I already gave you the answer here. The fragment, if the fragment is small, it ends up here at the bottom. If I loaded DNA plus a DNA binding protein, where would you expect the band to be in this lane? So that's my question for you, chat. Where is this band going to be if I load in this lane of the gel DNA plus DNA binding protein? So intact DNA will move slower. That is correct, A. If you have DNA that is not chopped up at all, like very big pieces of DNA, it'll migrate very, very slowly. Close to the start, exactly. So if we have DNA plus a DNA binding protein, we should expect it to move slower because it is functionally a much larger molecule. For those of you who are biology students, this is not a denaturing gel. This is a gel that keeps molecules intact. That's a thing only for the biology students here. So if that's the case, where do you expect DNA plus D-sub to be? if your hypothesis, the hypothesis that chat came up with, if you are correct, where do you expect this band to be located? DNA unbound down here, DNA plus DNA binding protein. If you are correct, where is this band in this lane? You got this, chat. This is a check for understanding here. Because y'all are, yeah, top, exactly. Thank you, Thomas. That is correct. So the hypothesis that chat collectively came up with, that it acts like a shield to protect the DNA, this is what we would expect. Even close to the start, exactly. So let's take a look at the 2019 paper. This is Jones et al. 2019, published in eLife, and this is what they found. So this is what an actual gel mobility shift assay looks like. And this up here is the ratio of DNA to dsub. So as we go from 0 to 6, we add more dsub. So this lane is no dsub. This is 6 molecules of dsub to whatever molecule of DNA. Is this what you would expect? Chat, if you were to just look at this DNA, look at this gel, would you assume that you were correct? From this data, does it look like dsub binds DNA? And be honest, chat. Be honest. Really, really, maybe, I don't know if I can blow it up a little bigger, if it's hard to see. It's a little hard to see. I'll make it a bit bigger. I'll make it a bit bigger. Make it a bit bigger for now. Does it look like the dsub binds DNA? Not really. Yeah. Your eyes don't deceive you. I can barely see the band that we expect to see. And that's kind of bizarre. At least I found this result to be a little concerning, because I, like you, had the hypothesis, oh, maybe dsub is binding DNA. But this experiment was done in purified DNA. If you were paying attention to the lecture, you know that in your cells, DNA comes in different forms, right? Somehow the DNA band is getting more faint. Yeah, that could be due to some of the dsub binding. So there's a little smear up here. It's barely visible. The DNA band is getting fainter. That's maybe because the dsub is binding and causing a little bit of a shift. But it's certainly not as strong as you would expect it to be, right? The cool thing about this paper is that dsub weakly binds purified DNA. But your cells don't have purified DNA. Your cells, the DNA in your cells is wrapped around histones. Right? Your DNA looks very different than purified DNA. Your DNA is supercoiled and wrapped into these tight little conformations. And so the authors decided to look at monosomes. So DNA plus histones. And now look at the data. Yeah, your dirty DNA, exactly. Dirty, dirty DNA. Monosomes, which are DNA plus histones. What does this look like to you, chat? How would you interpret this data? First, observe that the monosome is bigger than the DNA. This certainly makes sense because DNA, well, actually, I'm not going to have too much of conjecture, but compare purified DNA to the nucleosome DNA. So nucleosomes, again, it's your DNA wrapped around in histones. And when you add dsub, look what happens. Isn't that crazy? So only nucleosomes, so only your DNA wrapped around chromatin. Then you add 1.5 parts of dsub. This gets a little fainter. You start seeing a shift. 3 times as much dsub as DNA, you get a bigger shift. And all the way, when you hit the ratio of 6:1, that monosome is getting so tagged with dsub that it can barely travel through that gel. Isn't that cool? I think that's incredible. So chat, is your hypothesis correct? Well, so dsub binds binds monosomes. So it binds the DNA that looks like how it looks like in your chromosomes. Is this enough to say definitively that the way DSUP works is by acting like a shield to your DNA? Is this sufficient evidence that your hypothesis is correct, chat? Give you some time to stew on that. And this is a very, very advanced line of thinking, right? This is the kind of thing that I love to walk my students through, because the whole American education system wants teachers to teach critical thinking. This is critical thinking. You say no. Why is that? Why is this, in your perspective, in your opinion, why is this not enough evidence that DSUP protects your DNA from damage by shielding it? Thomas, I mean, it could be doing anything while it's bound to the DNA. That is true. Exactly. That is a great answer. We don't know what DSUP is doing as it's bound to the DNA. That's one part of the story. That is why in the education of science, when you go through classes, the thing that I would like for students to learn, preferably the earlier the better. So if you are in high school, I'm happy to challenge you in this kind of way. Even if you're not in high school and you don't typically get this kind of grilling, it's still important to think about the difference between something that is correlated and something that is causative. I want to say yes, but the fact that you're asking makes me think no. That's student meta, exactly. James, could it bind to the histones? It could be binding to the histones. I'm going to lump that into this idea of disambiguating correlation from causation. So Thomas has it right. Just because DSUP A protects DNA and B binds to DNA does not mean that DSUP protects DNA by binding to it. We need more proof. Does everybody kind of understand? I want to really make sure that this point is clear, because this is why I love teaching real, actual science. Is this idea of correlation versus causation making sense? I want to take a quick poll. Just because metagaming is totally fine. In fact, if you're pre-med, I highly recommend that you metagame entirely, right? Because that's what matters to you. But yeah, I'll repeat myself. Just because we know that DSUP protects DNA, and we know now that DSUP binds DNA that looks like how it looks like in your cell, that does not necessarily mean that DSUP is protecting your DNA by acting like a shield. It could be binding to your DNA by accident. So that's interesting. And so the beauty of biology, I think, is how do you disambiguate correlation from causation? How do you determine whether or not this is true? And it might not be very satisfying, but I don't have an answer. But what we can do is challenge chat to come up with a testable hypothesis or come up with a potential experiment to disambiguate this. Okay? So correlation is not causation. DSUP binds DNA. DSUP also prevents DNA damage. Yeah, A, you are right. It could just be hitching a ride. And Nizori, I will leave this up here to let it kind of sink in. So DNA binds a DSUP, this protein from the tardigrade that prevents it from taking damage via radiation. It binds DNA. It also prevents DNA damage. But it does not mean that DSUP prevents DNA damage by binding to it. This is the same kind of idea that I'll come up with another example. The number of US presidents is correlated with my age, for example. That doesn't mean that when we elect a new president, it causes me to get older, although during certain election cycles, it definitely causes me to feel older. But these two ideas happen to be happening at the same time. Now, something that is very important to get across is that when I call it baby's first critical thought, when you first get a hold of this idea that correlation is not causation, it's very tempting to just slap that across all of your scientific critiques, which, you know, I see on Reddit like all the time. Just because something's correlation doesn't mean that it's not useful. Just because something is not proved to be causative doesn't mean it's false. But we have to be very careful about what we ascertain to be the truth. Right? So, it's very tempting to believe that DSUP can shield DNA damage. It's very tempting. But we have to prove it. So chat, do you have an idea of how you would do this? And this is really challenging here if you're not a biology student. But if you're not a biology student, I think it's really fun to try. How could we test for DSUP binding? Or how could we test whether or not DSUP binding to DNA is required for DNA protection? If you're a biology student, if you are an undergraduate biology student, this is probably very easy for you. Well, not easy, but straightforward because you've practiced this level of thinking. But I would actually say that you can figure this out if you have troubleshooting experience. So I'm looking at the programmers in the room. Is that with your way of thinking as someone who troubleshoots software, you can do something similar here. Maybe try to recreate the setup. I usually don't. Okay, so so this actually connects to your next one, and remove the protein. So I'll say that that's part, you're thinking in the right way. But if you remove DSUP, we already know that you take massive damage via irradiation. Try to cut it in some way. Yeah, Dima, I'm liking the way you're thinking. Is there a way to disable binding? Yes. That's a great line of thinking. With a control, of course. Thank you. Always have to have controls up. So let's take a more careful thought a more careful direction where the chat is going, and you're all sniffing around the same idea. Disabling binding. How could we do that? I'm looking for abstract thought. Doesn't have to be concrete, just abstract ideas. How could we stop DSUP from binding to DNA? And I have two ideas. I mean, maybe there are more, but I have two. Two ideas. A, some protein. Yes. We'll call it, well, we'll even generalize. So protein is too specific. That's why I want to think very abstractly. So instead of some protein, let's think about it as some molecule, a blocker. Put something more attracting for it to bind with. Yes, Megacraft, that's great. That's a great idea. We'll call that a blocker. So you and A have the same kind of idea. You actually are all on what I consider to be the superior method of investigating this question. Very good. You did not fall into my trap, although this is a trap I specifically set out for biology students. So the biology students here did not fall for this trap, but I will explain the trap because it's important and cool to think about why, chat, your idea is better than the trap that I had put out. Okay? Very good. Let's follow along. So one idea that biologists can typically do when they want to see if things are necessary for DNA protection is to do something called a mutation. So one way, and I love all your answers, by the way. So disabling binding, you can do it in multiple ways. One way is to mutate the DSUP protein so it no longer binds DNA. And while I discuss this, think about what pros and cons could this lead to. Right, so we'll walk through this together. So wild-type DSUP. So this is the DSUP that you would find in the tardigrade, completely unmodified. You add your DNA. You shock it with UV light to damage it. And then you check the DNA damage. I'm just going to do this one for you because it's the easiest example. If you add the tardigrade protein that's supposed to protect the DNA and you check the DNA damage, you are going to get low DNA damage, right? That's not that bizarre. If you remove the DSUP and you add DNA and you check the DNA damage, this is another easy one. You added no protection, so this is high DNA damage. An altered D-sub might lose their protecting property. Yes, and more juice. You have to be careful about how you mutate the D-sub so it only kills the binding action. Yes, exactly. That is, you were getting a little bit ahead of me, but that's exactly correct. So mutant D-sub, you add DNA, you check DNA damage. What would you expect? Well, if the D-sub no longer binds to DNA, then we would expect high DNA damage, right? That if we mutated it to the point where it doesn't bind DNA, and our hypothesis is that it needs to bind DNA to protect it, then you would get high DNA damage. But more juice and now Thomas the thermonuclear bomb, you're all surrounding the correct point. There's a flaw here. How do you mutate D-sub so that it only disrupts the binding action and nothing else? Thomas, you're on the right track. This is really, really challenging, and it would be very hard to convince somebody that you could do this properly because, we just take a quick look back in the presentation. Usually mutations you want to do one at a time, but what the heck do you mutate in this spaghetti? Not entirely obvious. Very hard experiment to do. But this idea of mutating a protein, this is a very common approach to necessity studies, which is why it was the trap that I set out for the biology students, but nobody fell for it, which is good. Yeah, they didn't have, well, this one is 2019, so they do have CRISPR. But how do you know what to mutate to kill the binding activity? Very hard to know. So here's an alternative explanation, and this is the experiment that you all came up with, to use a blocker. Now, I don't know if a blocker exists. I don't know, I didn't read that deeply, but this is an idea that is worth thinking about because this gets around the ambiguity of idea number one. So instead of mutating D-sub, let's add something to block it. So again, wild type D-sub, you add DNA, you shoot it with UV light, you check DNA damage, you don't get DNA damage. If you don't have the protection, you add DNA, you shoot UV light at it, you get high DNA damage. What do you expect if you only add your blocker? So this is a control experiment. So A had suggested some protein. It doesn't have to be a protein. Maybe it's a small molecule. Who knows what this is? Just brute force it and mutate a bunch of D-sub? You could, but that is likely going to be a waste of grad school energy. This is a much cleaner experiment. If you add blocker alone, you would expect, yes, MegaCraft, you got it right, you would expect high DNA damage. Because the blocker, in an ideal world, this blocker's only job is to block D-sub from binding DNA. And not only do we expect it to be high damage, we expect it to be the same level of high damage as the no D-sub. If this blocker is an ideal blocker, right? Might not be the case, but if this blocker's job is truly just to block D-sub. So what if we added wild type D-sub and the blocker add DNA, check DNA damage? What would you see? This is the accumulation of your mind, chat. This is the experiment that you designed for us. What if the blocker turns off D-sub? That's what we want it to do. The blocker is supposed to turn off D-sub. So if the blocker turns off D-sub, what do we expect to see? Same damage as the two previous. Exactly. Exactly correct. This is clean. Unlike the previous experiment, this experiment is clean. It solves some issues that the previous experiment does not solve, right? Because with a mutation, it's hard to know whether or not you've just disrupted D-sub. But if you use a blocker, then you've left D-sub entirely intact. And if the blocker doesn't do anything funny on its own, then we have a very potentially satisfying answer. If you just stop D-sub from binding DNA, then you increase the DNA damage. So elegant, chef's kiss. Exactly. See, this is why I think experimental design can be beautiful, right? When we talk about taste in science, right? My taste is in these kinds of setups where, because biology, molecular biology, can have this kind of form, it's very satisfying to think through this logic puzzle, this argument, right? Because science is about argumentation, is that these two ideas were both equally valid in terms of they would both kind of answer the correct question. But idea two, the idea, chat, that I'm very proud that you came up with, this is the more elegant solution. This is beautiful. This does not leave that much room for interpretation. And that's why I think it's great. Is it perfect? Well, you could actually argue that the blocker alone could be doing something and it saturates high DNA damage, right? So let's say you add, no dsub, add DNA, check DNA damage, high DNA damage. Maybe that's the maximum amount that DNA damage can be at. You could argue that the blocker might also add to DNA damage, but it saturates, like you can't damage it anymore. But there's a way you can solve that too by just reducing the amount of UV that you use, and maybe you try to damage it as least as possible to try to tease out those differences. There are nuances when it comes to designing experiments like this. But I'm very impressed and very happy that you were all able to come up with this, because again, it's proof that we can start teaching critical thought earlier than undergrad. And, for me, I only started to learn to think this way in graduate school. If you apply this level of thought, this concept of necessity, sufficiency, this rigor in your ability to think, the earlier you start thinking like this, the better time you will have thinking, just in general. It doesn't have to be during research, right? I think if you can think like this is just like puzzle solving. And if you are a biology student or a biology student hopeful, getting to this point where you can logic out this puzzle, like this isn't even biology, this is just logic. If you practice this type of logic, I think you'll be a lot better off for it. Bit off topic, but how do you even check the level of DNA damage? Very good question, James. So one potential way you can do it is with an antibody towards something called gamma H2AX. So this is a marker for DNA damage that binds to the histones. So if you have an antibody to attach to the marker for DNA damage, you can actually count the number of places. Hold on, let me get that. Let me think for a second, because there are a couple of different ways you can calculate it. I believe that's correct. Yeah, so gamma H2AX is something that is a marker. It's a chemical marker that happens to your histones. You can get an antibody that detects that, and then so you can actually see how many spots your DNA broke, roughly. It's not really one-to-one, but you can kind of get a correlate of how much your DNA is broken. Assuming the damage is about fragmenting DNA, you can also use gel electrophoresis to measure how small the DNA fragments are after irradiation. That is a great idea as well. More juice, that's actually a technique called a comet chip, where you actually load in your DNA sample and then into a hole, and then you shoot electricity through that hole, and it drags out the small fragments of DNA like a comet. And so the longer the comet tail is, the more DNA damage you have. So it's a very good way to do it as well. Very, very cool. Okay, so I really want to hone this 101 series. I hope that you enjoyed this as because lecture can sometimes be like the professor just talks to you, and I think that's not, if you're a student, then that's fine, because you should be taking notes and actively studying. But if you're here because you like biology, or you like thinking about biology, or you like being challenged to do these logic puzzles, then I think this format really works for me. And so I want to craft each one of these 101 series in this kind of way, where, we're getting that kind of brain massage, right? So we started here with DNA at the beginning of the central dogma, and I want to then head to protein. I did RNA before, not in this style, but I don't want to look at RNA again. And then we'll talk about lipids, which are fats, and then we're going to get into some of the subcategories of cell molecular biology. Some of these will be collaborators with VTubers who are professionals in these areas, which will be kind of fun. You made me sweat. The good thing here is that I am not giving you a grade, but everybody in chat gets an A+ from me anyway, just for coming out and hanging out. I think it's really interesting. And the thing is again, to get on my soapbox, with American education especially, people lament not being able to teach critical thinking at a younger age. And I think that it's definitely hard, because think about how much education I had to have to do this. I didn't finish my PhD, but I did 6 years of it, right? Like, it's not easy to put together these stories. But the fact that there are either like people who only took an intro class, or people who are not biology professionals, or even high schoolers here who are not even done with their intro level biology, the fact that you can be led to thinking like a scientist means that this level of creativity and scientific thinking is possible. We can do it. And the thing is that not a lot of teachers are enabled or empowered to do this, right? Because, you know, you don't test critical thinking on your statewide exams, you know, the exams that we make you do, not we like me, but like the government makes you do. So when is a teacher supposed to fit in time to practice critical thinking? Also, where's the teacher getting taught critical thinking? That's another possibility as well. I'm not like crapping on our education system, but I'm saying that if there are people complaining about not being taught critical thinking earlier, there are good reasons why it's not implemented, because it is difficult. So I hope that like in this kind of lecture series, that idea, we can all practice that together. In my wildest imaginations, where I have numbers behind me, this could be a free place where anybody can practice that level of critical thinking. I feel a bit bad with throwing things and seeing what sticks most of the time. A lot of times since I did biology. I mean, the thing is, the act of throwing stuff to the wall is better than a lot of students. A lot of students who don't participate, who are not engaged, who are afraid to be wrong. And this is another soapbox that I get on all the time. Not on stream yet, so maybe this is the first time that I'm mentioning this, but I get being shy and not participating in class. Oh, like, I was also afraid of looking dumb in front of my peers. But the way that I worked around that was I tried to come up with one good question for my professor at the end of every lecture that forced me to throw my brain at the wall. And just by going through the motion of coming up with an idea, even if you were just guessing, just the idea that you were in it, that you had to come up with something, means that you improved yourself. This is not just me guessing, by the way. There is a study done by, I think, Harvard Education, where they compared between two different classes, identical in every way besides their structure. And in one of these classes, they implemented a paradigm known as active learning. So active learning is when activities are built and baked into the actual class. So activities can range from anywhere from like a clicker question to talking with a partner, discussing things, taking a short quiz. Classes that implemented active learning principles, the students had better information retention than their peers who did traditional lectures. So by using your actual brain, even if it is to guess, you are still using it, and that is sufficient to help you develop a mind for this kind of stuff. AP exams can be a mixed bag on critical thinking. Yeah. AP exams for me have been like a real, like a very long time ago. I actually have a shocking revelation. I actually never took AP biology. I never took the exam. I was so bad at memorization that I felt very confident that I was not going to be able to get a 5. But I love biology. And so I'm saying this because just because you're bad at memorizing things doesn't mean you can't be a biologist. It just means you have to work twice as hard. And the thing is that you have to know everything for the exam, and I talked about this on an earlier stream about moving past evaluation, like exam evaluation for evaluating students. You can't always get away with that. Like, it's fair. I didn't do as well as I wanted to in my biology classes, but I didn't love biology any less, and it didn't really ever discourage me from trying that, right? But yeah, the AP exams, I actually looked a little bit into the curriculum because I actually brought my AP biology textbook from my childhood home to my current living situation to see, to kind of like get an idea of like how I could do this lecture. And AP bio is like really hard. When I like self-administered some of the exam stuff, I was like, damn, like I don't remember any of this. But if you are like looking to get into biology, just know that like after you're done with grad school, you don't have to have anything memorized. you can always refresh. Like, no one's going to test you. You can always refresh on the biology that you need to use. But yeah, yeah, it was really fun. So I guess we'll head over to a little bit of chatting. Let's go ahead and get back to this screen. Be Big Five for a bit. We'll chat for a bit. Some reflections. Yeah, for me, memorization only works for things that I like. I'm not interested in spend 1 hour memorizing a paragraph. Am I still live? It says OBS for me is disconnected. I hope I'm still live. Okay, OBS is back. Lunch is over for me, so I got to get back to work. But this was fun. Thank you so much for hanging out. Okay, great. I'm back. Yeah, OBS kind of crapped out on me. I will be getting a actual computer. So the days of the Phi Top are quickly coming to a close. I will be a real streamer with an actual computer tower and not pushing my laptop to the brink. Potato PC, not even potato PC, potato laptop. This poor thing has been through so much. All right, so is there anything that I wanted to chat about, stuff that happened to me recently? I guess I could. So at my current job, this is a little behind the scenes. Actually, should I talk about that? Maybe not. I'll say that, like, it's an interesting perspective to have as an educator for people who are 18 plus, because by the time you get to college, you are expected. I have the expectation that by the time you get to college, you are an actual adult. And the cutoff for adulthood is kind of arbitrary in a kind of way, where, like, you know, when you're like 12, you know, if you are heading into a dark place, then your teacher has the authority to do things to try to help you out. But I noticed that as a teacher of adults, legally, that you can't really do that much. So I guess to address the younger folk in chat, if you are bombing, and I said this before, but it keeps on happening. It keeps on happening, chat. It frustrates me. If you find yourself in a difficult academic situation, use all the resources you can use to get help, because there's only so much that the staff can do for you. And at my university, we actually do a lot. Like, we go above and beyond. We have so many faculty per student that it's nuts. But make sure that you have the humility and the comfort to get help if you need it. Just put it that. 18-year-olds are only adults on paper. Yeah. That's why I find it hard to want to. So, I mean, the VTubing space, the age range is quite large. I find it difficult to want to collab with people who are very, very young. That's just my personal line, because I don't know, some VTubers out there are the age of my students, and I can't imagine collabing with one of my students. When I first found out your channel, I got scared because I knew you knew more than me, and for some reason it made me stressed out. You might want to talk to somebody about that, because that's going to happen a lot in your schooling years. I grew up in a very competitive high school environment, and I learned from a very young age that I was never going to be the person that knew the most in any room, and eventually just become okay with that, but it did screw me up for a long time. I've heard the brain finishes cooking at 26. I think that is a common misconception. I think that's a common misconception. I think that's been debunked, but I'm not entirely sure. It might be interesting to talk to a certain psychologist about how true that is. Sophie Yumi, welcome to the stream. It's hard to swallow that juvenile pride of being one of the best in high school to being a regular person and search for help, but it's worth it. Yes, I completely agree. As someone who has been put in my place very early on in my academic career, it's just not worth it. But I think that it's easy for me to say get help, because essentially what I'm saying is that you are now somebody else's not problem, but like it pushes the responsibility to somebody like a therapist or somebody like that. But like, yeah, that juvenile pride. I mean, Dr. K has a whole series of like gifted child syndrome, and as someone who could have turned out that way, I do feel a lot of sympathy for that kind of struggle. Maybe we should move adulthood to 26. I think that, like, I mean, you gotta draw the line somewhere. That's true, but I think once you are responsible for putting food into your own mouth for a little bit of time, and you are responsible for paying some of your own bills, and you've done that for a year, I think that's when adulthood could be where you could draw that line for adulthood. You could. It might not be the best or only metric by which you consider somebody an adult, but I feel that unless you have struggled with existing for a bit, you know, I grew up very privileged, which is why that's my perspective, where I didn't have to fight that hard to survive early on in my life. But having a job for a while, or being in grad school where you are still, you know, providing for yourself, that to me, I think fixed a lot of things in a certain way. I think it literally depends. Maturity depends in a very big extent in your environment. 18-year-olds are kids because they're still in school until 18. Yeah, exactly. Like there's still, you don't have a lot of autonomy at 18, especially if you're in a city and you, well, I shouldn't say that, because in a city you still have access to public transportation, but like having an expensive thing like a car for a while that can teach responsibility, like that kind of thing, you know. And of course everybody hits those milestones at different times, so it's really hard to draw that kind of line. You can also see the change when they spend a few years in uni and when they spend a few years working. Exactly. Like the work, I think, is really, really useful, because when you work, you have less time for being stupid or acting in a stupid way. Not to judge people's life decisions, but there are things that objectively that people regret. I don't know. I've been watching a lot of Caleb Hammer, so maybe this is why it's been at the top of my head. You know, the YouTuber who screams at people about their finances. I'm not going to talk about that channel too much on my channel, but I do watch it, and sometimes my jaw drops at you know, Caleb's content. 18 is good for most legal things because you have to leave your parents at some point. The way we gradually become adults is something nice about our society. Yeah, there is a sort of natural handoff, I think. In fact, I think going to undergraduate, going to college is a bit of a nicer transition, but like, as opposed to going from 18 and then immediately having to put food in your own mouth. That's tough, especially, you know, I don't want to talk about current events too much, but you know, it's hard. It's hard out there. Gen Z, I see you. I acknowledge you. It's hard out there. I will afford you this kindness. Like I acknowledge it. And college can be a way to try to equalize, stabilize, but it's only, you know, it is better used if you are proactive in your own well-being, which could mean a lot of different things for a lot of different people, you know, but reaching out and getting help is better than not, because, you know, I've seen students who have made it very late into their academic career without reaching out, and it's like a lot of money. University isn't cheap, which is why, like, you know, if you were a teacher of children, then you can do things like physically move them to a different classroom, where maybe their needs are better handled. But if you're an adult and you're bombing and you keep on paying tuition, you know, The university isn't going to say, hey, stop giving us money. You'd have to be really bombing if the university turns down your tuition money, right? They're not really incentivized to make you leave. So you have to be proactive in your own well-being. I think it's more like spectrum and 18 is the middle point. I think I like that. I think 18 is like the midpoint of their transition to being an adult. At least, again, because I want to be very mindful of the fact that some people have to like grind from when they're very young. I think if you had to grind for your own survival, that like you take on more of that adultness, that adult quality a lot earlier. Me, you know, undergraduate followed by graduate school. I mean, yeah, graduate school for me was a job. It still wasn't like a real job in the way that we classically think of a real job, because in a real job, and I'm sure that, you know, our friends in tech here can attest to, is that you have to deal with the possibility of being laid off. And I think that idea that you could be kicked out, that's a sobering thought. And I have done my best to exist in a way where I do not have to worry about that. Hello, Dan Doodle. Welcome to the stream. We're in the just chatting section here. Just finished our DNA 101 series. Gonna be honest here, I have no idea what a chromosome is. Never got told or taught. I had seen that you're a middle schooler. Don't worry, you'll learn all about that in biology. It's kind of... yeah, so the content that I make is not necessarily geared towards people who are pre-high school. Even my YouTube videos, I don't think I make any content that is appropriate for anybody who hasn't taken at least an introductory biology class in middle school. So I would not be too hard on yourself if a lot of what I say comes out as gobbledygook, because my content assumes that you have bits and pieces of biology from when you first took it, maybe in high school. But yeah, I like to think that I would have very low constitution out there in the real world, which is why academia is so cozy and cushy and nice. There are problems, of course, but I wouldn't trade my job in for the world. Gonna methylate the entirety of my genome for vibes. Very similar approach, potentially, to the DIY biologist I discussed in my previous stream. Just for the vibes, just for the kick. Just turn off all your genes. No need to worry about any of that. I think methylation would be a very interesting topic to cover in terms of what I cover here, especially because there was a talk that I had heard in graduate school about early life stressors and how that can change the expression of your DNA through a field called epigenetics. The issue about talking about epigenetics is that there's so much misinformation about epigenetics out there that I'm not really comfortable with talking about it, at least for now. So I'm sorry to get your hopes up, Dan Doodle, but like, it's very easy to blame certain circumstances about one's life on the idea of epigenetics. So for those of you who don't know, epigenetics is a field of genetics that analyzes how there are heritable traits that you can get from your parents that are not coded in your A, G, C's, and T's. So yeah, for this like 101 level, I'm not going to do a deep thing on it, but basically one way that your genes can be modified is through a process called methylation. So this is basically what happens when a methyl group is stuck onto your DNA, basically. And if you stick that methyl group on top of your DNA polymerase, or polymerase enzymes, I just speak broadly here, are going to run up to that methylation and kind of bonk off. So methylation is a great way to turn off the expression of certain genes. And the thing about that is that you can inherit that methylation pattern from your parents. So if your parents has, you know, a gene turned off by methylation, you can have that gene turned off as well. So it's something that you can inherit, and it's not coded in the actual. I like what somebody had said earlier. It's like in the metadata of the code, right? The code is A, G, C, and T, but the metadata is on or off. I could have butchered that, but in any case, what that leads researchers to find is that certain stressors in the environment can cause heritable methylation patterns that can affect you growing up, and it's not something that's hard coded by your DNA, but inherited via extra DNA stuff, I'll say. And what I've been seeing is that there has been a bit of doomerism about methylation, I think, which is half justified, but it's not sufficient. Like, there's this idea of biological essentialism, which is that, oh, I am this way because of my biology, and I'll never be able to change because I'm built this way. And biological essentialism is a very dangerous psychological space to get stuck in. So, if you are prone to blaming your biology for the way that you operate, methylation and the study of epigenetics will not help you get out of that situation. That's not to say that epigenetics doesn't play a role in your health. It definitely does, but the reason why I'm hesitant to talk at length about it is because people can get the idea that their biology is written in stone, that the way that people operate is written in stone. And certainly there are aspects of your biology that are written in stone, but it may cause people to underestimate how the impact that they can have on their health via their choices. So just want to be careful there. What about embryology? Yeah, so if you look here, I guess I moved away from my lecture screen. Lecture number 9 would be cell and developmental biology. So eventually, much later into this lecture series, I would like to cover some developmental biology, like embryology. Because here's the thing, I did very poorly in cell and developmental biology, right? In graduate school, that was my worst class because the biology of how sperm plus egg equals zygote and how zygote becomes full human, that's not easy. Learning how that happens is not easy. It is very difficult. There are a lot of things to learn. But I also find that even though it's so crazy difficult, for me personally, because my memory is so bad, the orchestra by which you go from single cell to full baby is insanely interesting and insanely cool. And I would love to cover that very soon in the future when I do get to that intro lecture. I'm curious as to how methylation could be inherited or permanent, since from my knowledge, a gene has info for activation and the RNA it makes. That's a good question, and I'm not prepared to answer that in any kind of depth as of right now. This whole time, I've been all like, what is going on here about epigenetics? Hopefully that brief explanation helps you just a little bit. Trophoblastic hyperplasia in ovarian cysts due to 2x maternal genes and 2x paternal genes. Like, it's cool how it links to epigenetic importance. Just want to peel back on that, Dan Doodle. Part of why there are certain topics I won't cover in videos is because I want to be very careful, and I screwed up in the last stream. I'm hesitant to call that kind of thing as cool. I just want to be careful with that kind of language. I can see that the mechanism behind certain biological phenomena are cool, but yeah, causing certain biology things cool, I want to be very careful with that. Yeah, so like looking ahead, I'm very excited to cover some of these things and to like be able to refresh myself on how some of these work. I have an exam on embryology this week. Maybe you could teach me a thing or two because I've forgotten almost all of it. It's very, very difficult. Can you cover how T and B lymphocytes mutate their DNA? Not at the top of my head, but maybe in the future, because that is very interesting. I believe I already did, actually, in one of my streams, although I can't remember which one it is. Yeah, it's awful. I just mean for a theoretical standpoint. Yeah, but I would like to be disciplined in how I call things like that and be mindful. Just because, yeah, it's easy to get lost in the sauce when you love biology so much and some of these don't affect you. But just like to practice that kind of mindfulness. This is insanely off topic, but where's Toxo? Toxo's at work. Yeah. I have summer vacation because I love my job. My job is great and I get time off. But yeah, so I am on the EST side of things. So, you know, so I can report that Toxo has a fan out there. Fan club, yeah, Toxo fan club. Get some Toxo emojis in chat. But yeah, so I've been talking to some VTubers behind the scenes about collabing, and I'm excited to bring you those collabs. I have one coming up on Friday, actually, with a clam VTuber called Spolex, and we're going to be talking about antimicrobial peptides, something that your body makes that kills bacteria. So it's going to be very fun, and I'd love to reach out and collaborate with VTubers in an adjacent space to me because I love talking about stuff like this. Would I ever do plant stuff? I did a plant stream with my fellow VTheorem member, Nepenthes VT. You can find that in my live tab. So I did mycoplasma infection in plants. So mycoplasma are tiny bacteria that are on the border of what we'd consider to be alive. And I talked at length with her about plant immunity and plant pathogenicity. I would certainly be, you're not the only person to ask for plant pathogens. I might do one in the future. From what I've read, it's mostly chemical warfare, which is not, I would maybe not do it as a video because I feel like with mammalian biology, the immune system stories are a lot more complex because mammalian immune systems are like wild. That's not to say plant immune systems aren't cool, but it's more like there's less of that story, the like immuno-evasion kind of story. It's more like they make this chemical which does this thing and that's cool. So maybe for a stream. I study in French, so nomenclature will be hard. Oof, that's rough. Yeah. I always admire the graduate students who come to the US and have to learn English to participate because there are English proficiency tests for the universities that I've been a part of. And like I'm, you know, arguably fluent in one language, much less than fluent in others. And to be able to do science in English in a language that you didn't grow up with, that's tough. So I have mad respect for people who study in America, especially from different countries with different languages. Wish I had access to the fire emojis. If I had my way, right? If I had my way, I would be doing this as a hobby forever. And certainly when you get into a financial position where you can have membership, they're all yours. Yeah, I truly think like the list, I've even stopped adding things to the list because the list of topics to cover was just getting overwhelming. Like I really will die before I finish all the content to make. There's years to go through. To be honest, English is the easiest language to learn because you can find almost everything in English. That's true. It's a very widely used language, but even I think English is really dumb. I think there are much more efficient languages with fewer rules or exceptions. Just as a method of communication, I find English to be very, very... I don't envy, you know, not being in the lingua franca or whatever it's called, the like universal language. Don't envy it at all. I hate English tenses. There are so many. Yes, there are a ridiculous number of English tenses, and I apologize for them. I'm not really a representative for the English language, but I do apologize. My language only has 3. Yeah. I know that there are a lot of other languages where the tenses are definitely not as bad, and conjugation is also quite annoying. As someone that speaks Spanish, kind of speaks English, and is learning Japanese, everyone has their own weird things. If a language has a long history, it has many rules. That's not, I find that that's maybe not, okay, so I guess to address that in order. Everyone has its own weird things. That is true. And I think, MegaCraft, you're being very generous to English, because English is my first language. And when I compare it to a language like Mandarin, Mandarin, the writing is really hard, but the grammar is really not bad at all. It's quite simple. If a language has a long history, it has many rules. And that's kind of what I'm getting at. Mandarin, the grammar is maybe the simplest grammar I've ever seen. You specify, if, for example, tenses. There's not really a conjugation. You just say when you did something. So if you wanted to say, like, yesterday I did something, in Mandarin you would just say, yesterday it happened, or something like that. You don't need to conjugate in Mandarin, and that takes a huge chunk off of your mind for grammar. Notehead, welcome to the stream. Finally, a bio course I can take online. I do say this, and at this point, like, I make this disclaimer every time. This is not a substitute for your actual class. This is more for fun and to exercise your brain. I haven't learned Chinese, but have you saw how Japanese enumerate things? They have different words for one depending on the shape of the thing. That is true. That also happens in Mandarin, and that's a huge pain in the ass. That is the bad thing, I think, about Mandarin, is counting. But I would trade counting for tenses like past perfect, imperfect. I would trade that. I would easily trade that. And the fact that, like, English, you have a different word for every month, but in certain Asiatic languages, you just say which month it is by number, which is very useful. My native language is a mix of mostly Arabic, Berber, French, and Spanish. Gosh, so many languages. Languages are cool. It's one of the incentives for a very high level of membership, which, was a stretch goal, because I was just like learning, and if I got paid to learn, I will learn anything. So if you want to if you want to test my brain on how fast I can learn a language, there is that membership tier for that kind of thing. Let's say English for fine in the chat. That's very kind of you. I don't mind the short bit of banter, just because we are talking about language at this point. I find languages to be interesting because it's a different kind of flexibility that you require. I think what is underestimated in the pedagogy of language is the role of play. So when I was a kid, I was shamed for not being able to speak Mandarin very well, and it made learning Mandarin not fun. And when learning Mandarin is not fun, it sucks. And then, like, you just don't learn it because it's embarrassing. So that's a kind of collective, what that reveals is two things. I think that's a collective error on the part of the previous generation on their methodology for teaching language. And B, it kind of delineates between people who are maybe sensitive about their egos versus people who are less sensitive, we'll say. So for me, I didn't realize, because for me, I don't mind admitting I'm wrong a lot, but I've come to realize in my reflection that I do have, like, a pretty big ego. It actually is kind of easy to hurt my ego in a fun way. There are not a lot of instances where my ego is hurt, but when my ego is hurt, it can cut pretty deep. And so I think that, reflecting on my past, I do have learning related anxiety, and part of that can be traced back to being shamed for not being able to learn Mandarin very well. It's pretty interesting. By the try guy, exactly. I would like to be paid to be a try guy, but for learning instead of, like, I don't know, baking cakes. The brain learns languages faster at a young age, so your native language determines a lot. Yeah, my native language is English, but I did learn Mandarin, like, before the critical period, like 12 or whatever. It's just, I did not like getting negative feedback with regards to learning. And this is kind of an interesting dovetail, because I talked about this on a previous stream, but I tend to only give negative feedback in my writing because students tend to legislate when they are given positive feedback. But, upon chatting with chat, I could maybe pull out the occasional positive feedback, the occasional you should do this again. But I would have to think very hard as to what to actually reinforce, because, you know, when you deal with students, especially students who want to go to medical school, they can legislate you on every single thing, and it makes your life a good deal more complicated. Of course, neutrophils are the only head. Yeah, big head pun. Yeah, that's true. I do have a big head. Selectively. It's really interesting because there's a lot of, like, not invulnerability, but I think I have a pretty thick skin with regards to certain aspects of my intelligence. But if I'm made to feel incompetent, it does, like, honestly hurt. Although, again, those situations don't really happen all that often. Especially not nowadays, where, like, you know, I'm not comparing myself to anybody anymore. Well, I compare myself to my fellow instructors because I admire my fellow instructors, and, you know, I just started teaching at this level, so there's a lot to learn about how to teach, actually, because that's something that I have been continually refining for almost, like, for probably a little less than half my life, I think. Instead of reinforcement, you can give a compliment if someone generally improves. Yeah, I mean, I want to. But then what I don't want to hear is, oh, you said that I improved. How come I did poorly in this assignment? And it's like, well, you did, but then you sucked again. It's a tricky thing, I think. Very, very tricky. Yeah, that's the kind of legislation I'm talking about, because they'll want every point back. And if you let them think that they're good, they're going to think that they're good. And then they think that they can't be bad. And I need them to think that they can be bad. Yeah. Please don't take it bad. We had a bad experience with foreign languages and side race trade. Megacraft, speak more of that. I missed that. That must be... I want in on the joke. Very interesting. Spanish is a really beautiful language. I'm happy to learn it. I found that when I was learning Spanish in school, that it felt like it was easier than English. Now, I can't speak any of it now, but when I was learning it, I was like, this is remarkably consistent. There are remarkably few exceptions to the rules that I learned, which was such a refresher having come from English. Just seeing non-English speakers struggle with English, I'm embarrassed by English. I'm like, damn, you're right, that does make no sense. But yeah, it's I know I return to the topic a lot in terms of, like, student feedback, but it's my job, and that's the thing that I do a lot most of the day, most of my days, right? But I think, like, I hope it's useful, at least for the students, to hear from the instructor's side. This is a very funny conversation I had with my primary care doctor, because, you know, some of you may know that I had shingles at a very young age as someone who's gotten shingles. And part of that was just, like, last semester got really bad with grading. And in that window of time, the grading, like, I had incredibly short turnaround time for so many students, and it probably affected my immune system to the point where I had a shingles outbreak. So for those of you who don't know, shingles is a reoccurrence of the chickenpox if you got chickenpox as a kid, which, if you are young, you should not get, because hopefully you are vaccinated. We love vaccines on this channel. IDK, if people would be comfortable with me saying it publicly, yeah, you can absolutely DM me. That'd be kind of fun. What was I gonna say? Yeah, I got shingles. So chickenpox is really interesting because you get chickenpox, you itch a lot, you get a rash, right? It infects your skin, but then it hides in your nervous system, and it hides for years until your immune system is weakened. And then when it's weakened, it can reoccur, and then you get a painful rash. And so I'd been joking around with my primary care doctor, and I was telling him the story of, like, dang, you know, like, the grading was really hard, and, like, I was working hours in a very short amount of time to try to get all the good feedback back, because I write a lot. I give a lot of feedback. I try to be as helpful as possible to my students. And he had said, oh, it's like that meme of, like, Everybody asks what my grade is. Nobody asks how the grader is. And I'm like, that's true. That's so true, fam. Like, nobody, like, students complain all the time. Oh, there's too much homework. We have too much to do. The class is too hard. No one's asking, dude, the grader is suffering because there's so much to grade. Like, do you, like, I just want to say to those students, like, do you think I grade this because it's fun? Like, we give you this amount of work because you paid for this level of education. And it is in our expertise of years of, like, I mean, my fellow instructors have years. I just started. But through the collective wisdom of how this works, through trial and error, we have refined this class to a point where this is the correct amount of work that you should do if you want to be competitive. Which is a sucky way of saying that, but like, hey, that's what you pay your university for. Yeah, it's bad. Because maybe they think that we have TAs grade the homework, but no, the TAs don't grade the homework. I grade the homework, right? And that's something that has to go back on a very short timetable. So, you know, if you're out there, you're an undergraduate student, you're about to start college, whatever, you know, keep in mind that, like, the job doesn't end for your teacher at 9 to 5. Sometimes they have homework, and it's your homework. And the better you do on your homework, the happier your teacher will be, right? Because I think when you are in school, pre-college, you don't really get punished for being inefficient. You don't really get punished for being inefficient. Like, I was guilty of this too, especially in middle school. I used to write long essays, long papers for all of my work because, if you couldn't tell, I like talking. I like feeling eloquent. I like orating. I like communicating. And that comes across in my writing. So, you know, I like saying things out loud, but my phi scripts are scripts because I like writing. And it took grad school to really punish me for writing too much. And at the undergrad stage, I think a lot of students are still stuck in the mindset of, oh, if I write more, then I will do better. To some degree, that is true. There was an influential paper out there that showed that the longer your essay was for the SAT, the standardized aptitude test that you take in the US, the higher your grade would be, controlling for other factors. So to some extent, the length of your assignment can influence, to a certain degree, can influence whether or not a grader thinks that you're actually any good. However, at the university level, if you're at a good university and your teachers get paid decently to care, you get punished for being too wordy. In the real world, when you apply for grants, there are word limitations to these grants. And so one of the most famous of these grants is the NSF grant, the National Science Foundation grant. So if you are a student, if you are an early undergraduate or a high schooler, you should be thinking about this grant if you want a career in science. It's a 500-word grant, 500 words, a 500-word research proposal for your own unique idea. And you won't necessarily get the funding to do your idea, but the NSF challenges students who are thinking like scientists at an early career stage, and they want to support them if they show that they have the potential. It's so interesting because, like, there's a lot of anime-esque drama in an academic career, right? You can imagine getting grants as a, like, tournament. Like, you are trying to refine your craft to the point where you win. It's a competition, for real. And so, if you are capable of producing intelligent thought within 500 words, that makes your application much more likely to be accepted, and you'll get this fancy, very prestigious NSF grant. So that's just a warning if you're still in high school. You should be mindful of how much you are writing. See if you can write the same thing but in fewer words and not lose any clarity. That's a very useful skill to have, and if you do so, your future graders will thank me. The more you write, the more mistakes you can make. Exactly. Like, you just say more wrong things the more you put on the table. But also, You know, that kind of thing of like the longer your thing is, the more intelligent you come across, that only works at to a certain point. When you get to my level, if you are unable to communicate your idea succinctly, that actually makes you look worse. It makes you look, I hesitate to use the word, but if we're talking about first impressions, it makes it look like you can't string together an efficient thought. It sounds like, because when you write, writing is a reflection of your thought process, right? It takes some skill to make sure that your writing reflects how sharp your brain is. And if you get good at writing, it means you're translating directly from your clear line of thought to your pen. But if you don't have that clear connection between your brain and your pen, then you can come off across as if you don't know what you're talking about, and that's very bad. So take some writing classes. You'll thank me. It's very important. All right. So I think I'm going to call it there for this particular stream. Thank you all for coming to the first of the 101 lecture series. I am going to be doing a journal club with Spolax the Clam. We're going to be talking about antimicrobial peptides. These are peptides, short proteins that immune system cells make that kill bacteria. It's going to be really cool. We're going to show you how they found out how these antimicrobial peptides work, and there are going to be some pretty sick images for us to see as well. Yeah. So looking forward to that collab as well as the rest of this 101 series. There will be no stream next week because I will be away, but then we should be back to our regularly scheduled, roughly weekly streams as I plow through this Prion video. Alrighty then. Bye, y'all. It was a lot of fun. See you next time.